All right, good morning, people. How are we today? Okay, well, let's get right into it. The age-old theory of evolution. Now, what this refers to is the change and adaptations that species go through over time. While it is a well-supported theory, it is simultaneously considered to be a fact by the scientific community. Now, in the intricate concept of evolutionary biology, the idea of missing links is a challenging topic that numerous researchers have tried to decipher over time. Now, there are two types. We have microevolution, which are adaptive changes that occur to species over a short period of time. Now, this is a definition that we are all taught as kids. However, the lesser-known macroevolution, this is the one that occurs over more long-term time spans and generally utilized when branching out lineages. Now, there are many ways to delve deeper into this topic. We have neural evolution, the evolution of the human brain across the years. We have ecological evolution, as well as avian radiation. However, the work I had assigned to you guys over the weekend has to do with the evolution of cells. So on today's agenda, we have eukaryotes versus prokaryotes, endosymbiosis, and archaea. Who would like to get us started off and tell us how this all ties together?
Gaining insight from Canera et al. In structural properties of promoters, similarities and differences between prokaryotes and eukaryotes. While eukaryotes evolved from prokaryotes, eukaryotic and prokaryotic organisms differ significantly in terms of their cellular structure, organization, and genetic makeup. Eukaryotic cells are larger and more complex, containing a true nucleus enclosed by a nuclear membrane and various membrane-bound organelles. Their genomes are typically larger and organized into multiple linear chromosomes. Eukaryotes can reproduce both sexually and asexually and exhibit diverse metabolic strategies. In contrast, prokaryotic cells are smaller and simpler, lacking a true nucleus and membrane-bound organelles, with their DNA located in a nucleoid region. Prokaryotic genomes are typically single circular DNA molecules, and their primary mode of reproduction is binary fission. Delving deeper, eukaryotes undergo transcription in the nucleus, followed by translation in the cytoplasm, while prokaryotes perform both processes in the same cellular component. Additionally, eukaryotic mRNA undergoes extensive post-transcriptional modifications, such as splicing and capping, contributing to the complexity of gene regulation. Prokaryotic mRNA, on the other hand, is typically transcribed as a continuous molecule and translated without significant modification. Furthermore, eukaryotic cells often possess a dynamic cytoskeleton made up of microtubules, microfilaments, and intermediate filaments, providing structural support and facilitating intracellular transport. Prokaryotic cells lack a cytoskeleton and rely on a simpler structural framework. In terms of cell division, eukaryotes primarily undergo mitosis, resulting in genetically identical daughter cells, whereas prokaryotic cells divide through binary fission, leading to the formation of genetically identical offspring. These fundamental differences highlight the diverse evolutionary paths and adaptations that have shaped the biology of eukaryotes and prokaryotes.
Okay, thank you very much, Aaryan. Does anyone have any questions for him? Oh, okay. Well, Wellington, do you want to come up and present yours now?
Endosymbiosis is the explained evolution of simple cellular life to complex eukaryotic life, and this outlines three major steps, which is an ancestral eukaryotic cell developing a membrane-bound nucleus. Secondly, that eukaryotic ancestor then engulfs an oxygen-utilizing protobacterium, which would eventually become the mitochondria. And then thirdly, some of those eukaryotic ancestors engulf a photosynthesizing cyanobacterium, which would then become the chloroplast. So for each of these steps, it's important to note that this is not necessarily a linear process and may have happened simultaneously or concurrently or even independently.
Okay, thank you, Wellington. Do we have any questions for him?
So what exactly is the mitochondria and the chloroplast?
So mitochondria and chloroplast's main function in the cell is in the synthesis of ATP as the main energy currency of life. So in the mitochondrial matrix, pyruvate is oxidized to acetyl-CoA, which is then used in the citric acid cycle to produce CO2, FADH, and NADH. FADH and NADH are then used in oxidative phosphorylation to generate a proton gradient across the mitochondrial membrane to synthesize ATP. Now, similarly, in chloroplasts, stacks of thylakoid membranes, known as grana, also take photons through two stages of electron transport to produce oxygen and NADPH. This also generates a protein gradient across the membrane, which then synthesizes ATP.
Nice, okay, thank you for the presentation, Wellington. Who's up next?
Okay, good morning everyone. I'm Khushi and I'll be talking about the origins of several proteins and structures in eukaryotes that have archaeal ancestry, specifically to Loki-Archaeotum, which is thought to bridge the evolutionary gap between prokaryotes and eukaryotes, as we can see here. Now, the cytoskeleton is a network of constantly moving fibers which extend throughout the cytoplasm of the eukaryotic cell. And similar to human skeletons, the cytoskeleton gives the cell its structure, and that enables it to carry out processes needed for cellular survival. Now, one of these fibers is a microfilament made out of actin, as we can see here. This is essential for processes such as cell division and phagocytosis, which allowed endosymbiosis to occur. Loki-Archaeotum contains 5 variations of actin, which share ancestry with eukaryotic actin. In fact, analysis found that these variations were more similar to eukaryotic actin than they were to other archaeal actin. This suggests that Loki-Archaeotum is a direct ancestor of eukaryotes.
Thank you very much, Khushi. Do we have any questions from the audience?
What kind of analysis was done to discover the evolutionary link?
Well, on the sample, quantitative PCR, metagenomic sequencing, shotgun sequencing, and binning were all used to obtain the genetic data from the samples, but the links themselves were found through phylogenetic analysis, which is the study of evolutionary relationships between organisms. It usually involves some statistics to make phylogenetic trees, which is what we have here. And yeah, that tells us the evolutionary link. Thank you.
Okay, thank you, Khushi. You can have a seat. Okay, now tell me, what was the point of all of this? Why is this all important?
The medical implications. Knowledge of cell evolution aids in understanding the molecular basis of diseases. Comparative studies across species can reveal conserved genetic pathways and structures, offering potential targets for medical interventions and inventions and drug development. However, on the other side, biotechnological applications, insights into cell evolution contribute to advancements in biotechnology. Understanding the genetic and molecular differences between organisms can be harnessed for the development of new technologies, such as genetic engineering and synthetic biology. The study of cell evolution is fundamental to our understanding of biology.
Okay, thank you everyone for your presentations today. We covered quite a bit. All the presenters, can we have a round of applause, please? All right, well, class dismissed. Good luck for your exams.